In this third lecture on the survey of herbicide mechanism of action, we are going to finish the herbicides inhibiting biopolymer synthesis, and then we'll cover uh, some of the plant growth regulators real briefly. So, as I mentioned, we talked about cellulose biosynthesis inhibitors, lipid biosynthesis inhibitors, amino acid biosynthesis inhibitors, and the third one after EPSPS and ALS as target is GS, the glutamine synthetase, which is the group 10 herbicides. And I put this one a little bit apart because it is an inhibitor of amino acid biosynthesis, but the way it kills the plant is actually not through the inhibition of amino acid biosynthesis, but we recently established in our lab that it acts through photosynthesis and causes accumulation of reactive oxygen species, and I wanted to treat that separately from the other amino acid biosynthesis inhibitors. And so first of all, it was first discovered in 1981 as a natural product, was commercialized in Canada in 1993 and then in the United States. It was discovered and produced by Bayer Crop Science, but now it actually belongs to BASF. It's a non-selective broad spectrum herbicide. It goes in the leaf through the post-emergence application, does not translocate. It requires quite a bit of rates, up to 1 kilogram active per hectare. It has no soil activity like glyphosate. If you go in the soil, it binds to the soil and has no activity at all. And the symptoms are very rapid. You get burned down of the tissue very quickly. And it's one of those herbicides that's very dependent. The efficacy is dependent on the environmental conditions, and the plants have to be pretty small. If the plants get to a certain size, these herbicides will not work really well. And so drought and cool temperatures reduce control by this herbicide. That's inhibitors of glutamine synthetase is glufosinate or Liberty. And you obtain typical symptoms like this of the burn down of the tissue that was exposed to the herbicide. So glutamine synthetase inhibits glutamine synthesis, and that's an enzyme called glutamine synthetase, and it's a two-step reaction. It takes glutamate, adds ATP to it, or uses ATP to add a phosphate group to it to make glutamyl phosphate, and then the transamination where the ammonia is incorporated into the glutamate to make glutamine. So it's a two-step reaction. And what's interesting is that glufosinate is this molecule right here, and in the plant it gets phosphorylated to this. And what's interesting is if you compare phosphorylated glufosinate to this intermediate here, the gamma glutamyl phosphate, it's actually very similar, and actually you can overlay them on top of each other and they are very, very similar, so that phosphorylated glufosinate binds and competes for the binding domain in the enzyme. It's a competitive and it's irreversible. It binds and is not released, so it's also called a suicide inhibitor. And we spent a lot of time working on this, but what we've discovered is that while indeed glufosinate inhibits the conversion of glutamate to glutamine over here, that step is actually toward the end of what's called photorespiration, which is a mechanism of removing toxic components that are made when plants use oxygen rather than CO2 as part of photosynthesis. And so when glufosinate inhibits glutamine synthetase at this step, there's a backup of these precursors, and that actually backs up photosynthesis, which is, the Z scheme we talked about with photosystem 2 and photosystem 1 inhibitors. Glufosinate backs up the whole system, and these excess electrons actually react with oxygen and form hydrogen peroxide, superoxides, and hydroxy radicals, which are very toxic reactive oxygen species that can react with membranes, and that causes lipid peroxidation. And so it's kind of complicated that an inhibitor of amino acid biosynthesis actually kills plant by the formation of ROS. And then you have very rapid desiccation of the tissue. Then other than these three enzymes involved in the synthesis of the building blocks of aromatic amino acids, branch chain amino acids, and glutamine, then there are herbicides that inhibit enzymes involved in protein assembly, like trafluralin. These are the group 3 and group 23 herbicides. What they do is they bind to macrotubules. Actually, they bind to the macrotubules, and they prevent the assembly of the macrotubule. Um, and of course, there's a whole lecture on this if you're interested. But essentially, macrotubules they're part of the cytoskeleton. They help cells have their structure. They also help cells in cell division. They help cells in cellulose biosynthesis. So they are very important molecules. And those inhibitors prevent the formation of macrotubules, and that basically stops plant growth. And these herbicides, such as the dinitroanilines, tend to be pre-emergence herbicides applied before the plant emerges from the soil. So they control small-seeded weeds, grasses, and broadleaf. And they can be used in large seeded crops because they have very little effect on large seeds plant. They inhibit mitosis very much like colchicine, but they are more effective in plants than colchicine would be. And they bind to tubulin, and by binding to tubulin, that prevents the assembly of the macrotubules. And as far as we know, they inhibit the polymerization of tubulin into the macrotubule. So that affects cell division in the roots mostly. So as the seed germinates, they actually don't grow very big and they barely come out of the ground, if any, and then they will die. They will not establish themselves. So then there are many other herbicides. There are plant growth regulators, and I will talk about this, but there's also inhibitors of serine threonine protein phosphatase by a product called endothol. There's inhibitors of dihydropteroid synthase by other products, and there's also a lot of herbicides that have remained unknown at the moment. So I don't want to cover those, but I will talk a little bit about plant growth regulators. So these herbicides act like a natural hormones. So the group O, actually the group 4 now, mimic the effect of auxin. So they are auxin mimics. They act just like auxin, but instead of the plant being able to regulate the amount of auxin that's there, when you put the herbicide, you overwhelm the system, and what normally is tightly controlled reactions catalyzed by auxin, 2,4-D and dicamba, this type of chemistry will bind to the same domain and actually lead to uncontrolled growth of plants. So typical symptoms will be epinasty. The plants will grow faster and then it will bend down because some of the meristems, they will bend. So that will be the symptoms of epinasty. The stems will swell and the cells will not divide correctly anymore. This is a typical example of amino pyrrolate on Serosium right here. So you see these plants are lighter, but also you can see the bend right here. Those leaves, those meristems bend down. And that's very typical of auxin mimics. They tend to be root absorbed, but there is some foliar absorption as well. They cause callus formation in the roots because they are growth regulators, so they mess up with the hormonal balance in the plant, and plants then will start making callus instead of making tissue. They can translocate to the growing points, so then there's effects up there. The roots tend to be swollen, and very often there's very root development, so the plants don't establish and they fall over. 2,4-D was actually the first herbicide ever discovered and commercialized, and turned out it was an auxin inhibitor or auxin mimic. It was the first selective herbicide, and it's used in all these situations, mostly on annual weeds, but also some perennial weeds, even though they don't work as well on perennials as they do on annual weeds. So this concludes our lectures. I just want to give a couple of plugs. Todd Gaines and I just finished teaching a herbicide selectivity and action course. And the next time we will teach it will be in fall 2022. So if you're interested in how herbicide works, how herbicide resistance evolved, controversial issues in weed science, how to do diagnostic of herbicide injury, why do herbicides work sometimes and sometimes they don't, why do they cause crop injury sometimes. We cover all of this in that course AB 509, which will be taught again in the fall of 2022. And next fall, I will be teaching my plant biochemistry in agriculture, which is a course that basically covers plant biochemistry. There's no other courses at CSU that talk about plant biochemistry, only dedicated to plant biochemistry. And we go from amino acids to proteins to the main steps of photosynthesis, carbon assimilation, ammonia assimilation, amino acid biosynthesis. But what's really interesting about this course is that a couple of things in addition to the content is that every topic I try to include a reason why this particular biochemical process is important for agriculture, where it be for crop production or plant insect interaction, disease resistance. I try to find applicable examples to make it very relevant to people like in plant science, in botany, ecology, crop science, weed science, but also horticulture. And even if people are interested in entomology, insects that feed on plants, this course would be relevant for you as well. And I'll be teaching this in fall of 2021. So this August, I will start teaching this. So this was a very quick overview of something that we talk a whole semester about. So I'm sure it was more information than you wanted. I will provide all of these lectures as well as PDFs, and Dr. Gaines will post those on Canvas for you. Thank you.